Hello, everyone. So, welcome back to the second session of the day. I hope you all enjoy the treasure hunt on Gather. I'm Roji, and I will be moderating this session. So, our first speaker of the session is Emily Farran from University of Surrey. Her work concerns spatial cognition abilities in typical and atypical developing children. The title of her talk today is Spatial Exploration Patterns and Predictors of Navigation Competence, Evidence from Typical Development, Down Syndrome, and Williams Syndrome. So I will hand over the stage to you, Emily.
Thank you. I'll just share my screen. Okay. So thank you for inviting me to talk to you today. So as the speaker said, I'm going to be talking about our data on exploration behaviours in typically developing children, in people with Down syndrome and people with Williams syndrome. So the reason that I'm interested in atypical development, so people with Williams syndrome and people with Down syndrome, is because, as we all know, navigation is absolutely critical for you to be independent. So I've just listed simple types of navigation that might be limiting for these people, which might mean that they can't be independent of their parents. It might mean that they're not able to join the workforce, for example. So this is the first group I'm going to talk about, people with Williams syndrome. So for those of you that aren't aware of Williams syndrome, it's a rare genetic disorder, has a prevalence of 1 in 25,000 births. This is a behavioural study, and so I'm not going to pretend to know a huge amount about the neural activation for navigation in Williams syndrome. There hasn't been any research in that area. But what I can say is that parts of the brain that are activated in navigation are atypical in Williams syndrome. In terms of their behaviour, if you look at their use of landmarks, their use of their ability to form route knowledge or configural knowledge, all of these show atypicalities. Particularly configural knowledge is something that is very hard for someone with Williams syndrome to achieve. They can achieve route knowledge through a lot of practice, but obviously these are fairly inflexible routes. So there's a real reliance on egocentric strategies. And recently, when we've been talking to parents of people with Williams syndrome, the majority of people with Williams syndrome, even as adults, aren't allowed to travel independently or choose not to travel independently. So people with Down syndrome, Down syndrome is a more common genetic disorder with a prevalence of 10 in 10,000 live births. Again, there are areas of the brain that are linked to navigation that are atypical in Down syndrome. There's also evidence of difficulty using landmarks, of poor route knowledge and poor configural knowledge. So again, people with Down syndrome rely mainly on route knowledge, and even then it takes a lot of practice to develop any route knowledge. There has been some more recent evidence which has shown low-level allocentric coding for some people with Down syndrome, and this was in a kind of open environment, a bit like a sort of visual search environment. There's been some research into map use in Down syndrome, which again has shown that there's limited benefit of using maps in this group. And again, we asked parents about independent travel for their sons and daughters, and the majority said that their son or daughter with Down syndrome did not travel independently. So I'm going to talk to you today about Project Ellstrad. This was a 5-year project across England and France, where we were interested in navigation in typically developing children, in Williams syndrome and in Down syndrome. So I'm going to talk to you about the study where we looked at exploration strategies. Just to give you a bit of a caveat, this study, this data that I'm going to present today is from, I think, 2015. And unfortunately, the postdoc on this project got sick, very sick at the very end of the project. And I'm sure my collaborators won't mind me saying that they've all since retired. So this paper and the study that I'm going to talk about today has been a real labour of love in terms of getting the data ready for analysis. And I'm really indebted to some fantastic reviewers of the paper that have really helped to shape the paper and make it a lot better. So I'm hoping for even more feedback today on this data. So I'm just going to define what I mean by my different terms here. So in the human literature, a lot of the time when people measure sort of spatial knowledge or measure kind of navigation in virtual towns, the things that are measured are spatial knowledge measures, so understanding whether people can learn a fixed route, whether they can develop a cognitive map or configural knowledge, so pointing between unseen areas of an environment, for example. What I'm going to talk to you today are the kind of encoding behaviours and the patterns of behaviour that are exhibited during the gaining of that spatial knowledge. So the kind of unconstrained exploration patterns which allow people to develop that spatial knowledge. So the kind of how as to how the spatial knowledge is developed. So in humans, there isn't a huge amount of research into exploration behaviours. So I'm going to present to you what I have found so far, but of course, if there is some more research that people are aware of, please let me know. So there are a couple of studies in the adult literature where they looked at people's exploration strategies, and they found that fewer revisits to the same place, higher diffusion, so this is a little bit like roaming entropy, so how much area you're able to cover, and fewer pauses is related to navigation success. And there's another study by Vietz et al. Where they looked at people's use of route strategies and orientation strategies. And what they found is when they asked people retrospectively to tell them how much they were allowed to explore in childhood, they found an association between people's exploration opportunities. So reduced exploration opportunities in childhood were associated with the use of a route strategy over an orientation strategy. So it might be that there's a connection here between exploration opportunities and sort of individual differences in adulthood. And we've been hearing quite a lot in the literature lately from Stephen Weisberg and the individual differences that we see in navigation strategies in terms of integrators and non-integrators and people who are generally just poor at navigating. There's also a study by Cornell where they asked children to take them to the furthest place they'd ever been to as an adult, and they had a range of ages from 6 to 12 years. And what they found developmentally, that as children got older, they used distant landmarks more, so they saw a lot more scanning of the environment, which is indicative of the development of configural knowledge or the use of configural information, and that they had better landmark choices. And this actually relates back to Kate's talk earlier. And so this evidence suggests that exploration is an important determinant of the development of spatial knowledge. So in this study, I'm going to talk to you mainly about the typically developing data, but I've also got slides on the Down syndrome and Williams syndrome groups as well. So we had a range of children who were typically developing from 5 to 11 years. So we had a nice span of chronological age. The Down syndrome and the Williams syndrome groups were aged from kind of early adolescence to adulthood. What you can see here as well is BPVS, so that's a measure of verbal ability. So, so this is British Picture Vocabulary Scale, and then RCPM is a measure of non-verbal cognition, so Raven's Colour Progressive Matrices. So you can see there's variation across the groups in there, but what we were trying to do is get a group of typically developing participants which were kind of showing the same range of abilities in our navigation and exploration measures as the Down syndrome and Williams syndrome group. So we had 2 studies. The first we've labelled free exploration. So this, if we're using Wiener's taxonomy, is measuring undirected wayfinding. So this is similar to if you take a child to a park that they've never been to before and they just want to explore where everything is in the environment. So we asked children to start at a home location. They had 3 minutes to explore this virtual environment, and they were told to learn as much as you can about where everything is in the town. So it was a desktop virtual reality situation, and they navigated using the arrow keys on the keyboard. They only saw the environment from a first-person perspective, and the blue bar at the top decreased as the trial went on. And there was just one trial of this. You can see here the overhead map of the environment. So we had 10 building blocks. We had 40 proximal or local landmarks, 4 around each of the building blocks. This is the home location, and then we had 3 distant landmarks as well. So we were interested in their behaviour when they were asked to explore. So we wanted to look at the number of areas that were visited. We wanted to look at their path length. We wanted to look at the number of pauses and the number of revisits. So revisits was a composite of when the path intersected or crossed over itself, and also when they made retraces. So this was when they went in a particular direction and then turned around and came back again. And these were chosen based on the adult literature. So the diffusion variable of ganglion is similar to number of areas visited. And this was, we divided this area into a matrix of 3 by 3 so they could get a score out of 9 in terms of areas. And then Munyon looked at pauses, and both Munyon and Gagnon looked at revisits. So you can see here some scatter plots. So we were interested developmentally as to how these behaviours changed. And what you can see here, if we're using a bomb for any corrected alpha, so 3 of these are significant. So the relationship between revids and chronological age is not significant. But what you can see is that the number of areas visited goes up with increasing age, the number of pauses goes down with increasing age, and the path length goes up with increasing age. So in this free exploration scenario, it seems that the optimal strategy for exploring is to visit more areas, to pause less, and to have a longer path length. We wanted to look at this in Williams syndrome and Down syndrome as well. So the typically developing data, you've seen this row already. But in Williams syndrome and Down syndrome, chronological age is not necessarily a very good indicator of ability. So whilst I have chronological age here, you can see that it isn't correlating with any of our exploration measures. So we included our non-verbal measure as a kind of measure of non-verbal mental age. But again, you can see it's not correlating with any of our exploration variables. So this suggests to us that we haven't really tapped into what is driving performance in the Williams syndrome and Down syndrome group. And it's likely some of the variables that we've measured in earlier points in Project Ellstrad. And Veronica Mafato yesterday talked about visuospatial working memory. So it could be that, it could be executive functions, for example, as well. What we did do is we wanted to look at the kind of level of ability of the Down syndrome and Williams syndrome group. So we split the typically developing group into 3 age groups, so 5 to 7-year-olds, 8 to 9-year-olds, and 10 to 11-year-olds. And what we were able to show was that for the Williams and Down syndrome group, their sort of level of ability in terms of exploration behaviours was similar to the younger children, so the 5 to 7 and 8 to 9-year-old groups. So moving on to our second study here. So this was exploration with a goal, and using Wiener's taxonomy, this taps into directed wayfinding. So in this study, participants started at the home square, and they were asked to locate 6 stars throughout the environment and return to home within 3 minutes. We wanted to assess their learning, so they had 5 trials. In each trial, the stars were in the same place so that they could start to learn the configuration of the environment and the best routes to collect those stars. We had 2 conditions. I'm not going to talk about those a huge amount today, but we had one standard condition, and we had a condition where there was an overhead map in the corner of the screen. And on the screen, they could see each of the buildings, they could see themselves in real time, but also they could see each star that they collected. So that would appear as a yellow dot as they'd collected it. So it kind of told them where they'd been before. Also, for both conditions, as they collected the stars, there was a kind of scoreboard along the side, so they could see how many they collected and how many more they needed to collect. So here's some of our data. What you can see here, both of these participants, they both collected 6 stars and returned to home within 3 minutes. So they both achieved the goal, but clearly you can see that this person is a lot more efficient than this person. Because we wanted our kind of navigation success measure to be pretty sensitive, we took into account the time it took them to return to home. So they had a maximum of 180 seconds, and this person did this in a lot less than 180 seconds and a quicker time than this person. So rather than using the number of stars as our navigation success variable, we used the time per star as a navigation success variable. So it was 180 seconds divided by the number of stars that they collected. And that's what I've plotted on this plot here. So this is just the typically developing data. And what you can see, these are the 5 trials along the x-axis and time per target on the y-axis. And you can see a linear effect of their training. And so the more they're familiar with the environment, then the more successful they are at navigating and getting those 6 stars in good time. You can also see here that the overhead map condition there was a significant main effect, such that there was stronger performance in the overhead map condition. So we were interested in determining how much our 4 exploration variables were predictive of that navigation success of the time per star. So what we looked at was that we used a multiple regression, and so we inputted our 4 navigation variables into the multiple regression. We also had a variable of target consistency. So this was a variable of how consistent their order in collecting the stars was from one trial to the next, so that we could get a measure as to how systematic people are being in the environment. So what you can see is over and above age and gender, our exploration variables and the target consistency score account for 41% of variance in navigation success, so in our time per target variable. And when we look at this at the individual predictors, the exploration variables that are predicting performance are number of pauses, a number of areas, also number of revisits, although the standardized beta for this is fairly low. Also target consistency, which you might expect, is also predicting navigation success. Now, in this environment, this data was across all 5 trials, so it's exploration cumulated across the 5 trials and navigation success across the 5 trials. And you could argue that it's difficult to separate the exploration experience from the spatial knowledge, because people might be getting spatial knowledge while they're exploring and feeding this back in. So we also looked at their performance in terms of exploration in trial 1. So in trial 1, the environment was completely unfamiliar to them. And so there's no way they had any spatial knowledge of the environment. And we looked at this in relation to their spatial knowledge, so the navigation success across the 5 trials. And again, you can see that after accounting for age and gender, 21% of variance is accounted for in navigation success. And the same 2 strong variables are coming through in terms of number of pauses and number of areas as being important for navigation success. So to summarize, exploration behaviors explain 41% of navigation success, and efficient exploration is related to having fewer pauses and visiting more areas. And there's some evidence as well that it's related to having fewer revisits. So the other thing we wanted to do, I was quite influenced by Stephen Weisberg's work on individual differences in navigation strategies, and I wanted to look at individual differences in exploration strategies. So I took a data-driven approach and used latent profile analysis, and what this does, it gets your data set and sort of works out where the homogeneous subgroups are. And doing this, it gave us 3 different profiles of people. So what you can see, so negative scores on time per target reflect very good navigation success. Positive scores reflect poorer navigation success. So we have 3 different levels of navigation success, so very good, medium, and not so good. What you can see that differentiates these profiles, it relates mainly to the pauses in the path length. So profiles 1 and 3 both have relatively low pauses, and profile 2 has relatively high pauses. So you could say profiles 1 and 2 are kind of more active strategies, and profile 2 is a cautious strategy. Then if we differentiate profile 1 and 3 from one another, you can look at the path lengths and see, oh, profile 1, they're fairly efficient, their path lengths are quite short. Profile 3, they're much less efficient because they have longer path lengths. Interestingly, profile 2, who are very poor at navigating, also have short path lengths. So there's 2 ways in which you can have short path lengths. One is if you just don't get very far, and 2 is if you're very efficient and have very good knowledge of the environment in order to create a short path length. So to summarize, those 3 profiles in terms of individual differences reflect active versus cautious strategies and more efficient explorers versus less efficient explorers. So to look at the Williams syndrome and the Down syndrome data, this is the time per target graph. I've split the typically developing group into 3 age groups here, so 5 to 7-year-olds, 8 to 9-year-olds, and 10 to 11-year-olds. What you can see is that every group is showing learning across the 5 trials, so their time per target is going down, so they're getting more successful. but there was an interaction such that the Down syndrome group showed less of a learning effect than the other groups. There's also a main effect that their ability overall was poorer than the other groups. The Williams syndrome group were similar to the 5 to 7 and the 8 to 9-year-old groups. I haven't plotted condition on here, but there was also an interaction between the standard and the overhead map condition, which showed that there was an advantage of the overhead map condition, but it was only for the typically developing older children, so the 8 to 9-year-old group and the 10 to 11-year-old group. So I haven't done a regression with the Williams and Downs group due to the participant numbers, but what you can see is some correlations here. So the typically developing data you've seen already, where we've shown that pauses and areas are very predictive of navigation success. In the Downs and the Williams syndrome group, we have some parallels with the typically developing group. So we have pauses in the Williams group, areas in the Down syndrome group, but path length seems to be an important, has an important association with navigation success in the Downs and Williams syndrome group. I think this simply reflects that whereas in the typically developing group, we had 2 reasons for short paths, being very efficient and being very poor. I think for the Downs and Williams syndrome group, we don't have that very efficient group, so there's more of a linear relationship. Here's our correlations of the data I've just shown. So for pauses, you can see this is significant for Williams syndrome and Down syndrome. For the Williams syndrome and the typically developing group, for the Down syndrome group, we've got a bit of a cloud here of the poor navigators. For number of areas visited, we see that that cloud is showing that the people who are not very good at navigating just don't move very far. They don't cover much of the area. Number of revisits, nothing was significant. For path length, you can see that the Down syndrome and the Williams syndrome group are showing a significant effect. But it seems to be that those with shorter path lengths are actually the people that are not very good at navigating. So we ran the latent profile analysis again. So this is with the full set of data. So the Downs, Williams syndrome, and typically developing. And what we found is we have 3 profiles which are similar to the ones that we had before, and then we have these 2 new profiles. This group looked fairly average on everything, although this is an average across those 3 groups, 2 of which are atypical. And then we have a very extreme group of people who are just really, really poor at navigating and don't get very far. The most interesting thing from this profile analysis is to look at the composition of each of the groups. So what's really interesting here, so profiles 1 and 2 are the new profiles, and you can see that they're majority Williams syndrome and Down syndrome. Profile 1 was the very poor group, and in fact, this is only 7 people who are Williams syndrome and Down syndrome participants. Profile 2, this was the very average group, and then profiles 3, 4, and 5 were the profiles that we saw before, and I've listed them at the bottom of the screen. What I think is really interesting to show here is there isn't something systematic in the Williams or Down syndrome group which sets them out as being different from these typically developing children. I mean, okay, the Williams and Downs are adults and we're comparing them to children, but even in the most efficient strategy, group, which is profile 4, we have Williams syndrome and Down syndrome participants. And I've just put a plot here of the kind of behaviour that we were seeing from the profile 1 people. So they just didn't get very far and just weren't very successful in collecting any targets. So to conclude, we've looked at different exploration behaviours and shown that it predicts navigation success. And this is even when exploration is in an unfamiliar environment, it's predicting success once that environment becomes familiar. In typically developing participants, stronger navigation is associated with fewer pauses and visiting more areas of the environment, and in some situations, fewer revisits. And this is consistently, visiting more areas is consistent with Gagnon's variable of diffusion. And this could relate to, it kind of aligns with roaming entropy, and Heller et al. Have talked about the sort of reward value of an environmental novelty of visiting more areas. The relationship with pauses is consistent with Munyon, and this suggestion of differences in strategies in terms of active and cautious strategies. For the Williams and Down syndrome group, path length was important. I think, as I said before, because people were with short path lengths it wasn't because they were efficient, it was because they were poor at navigating. And also visiting more areas and having fewer pauses, consistent with the typically developing group, was important for these two groups differentially. For the Down syndrome group, I think this was related to the ones that were really not visiting many areas at all, were very poor at navigating, and that seems to be quite specific to the Down syndrome group. For the Williams syndrome group, having fewer pauses seems to be similar to the typically developing group in terms of strategizing. This is my last slide. So we looked at individual differences. We found differences in active versus cautious strategies and coupled this with differences in efficiency. And I'm wondering whether having an active strategy and a short path length suggests that the spatial knowledge that you're developing is configural knowledge, and having an active strategy and long path length suggests the information that you're gathering is the knowledge you're gaining is to do with root knowledge. But this is something we're yet to see. So in terms of next steps, I'd like to do a battery of tasks where we included configural knowledge tasks and root knowledge tasks specifically. Also, I'm very aware that we've just been looking at exploration behaviours on their own, but it would be important in the next steps to also look at the contributions of other factors that we've talked about before, executive functions, for example, and exploration behaviours within the same model. So just to say thank you, and just to say that our findings highlight the importance of exploration behaviours as a vehicle to understanding the development of spatial knowledge. Thanks.
Thank you, Emily, for the exciting talk. So we already have questions coming in. The first question is from Janina. So have you looked into the strategies participants have used during exploration? Did you, for example, find any systematic patterns in the trajectories, such as lawnmower patterns or people first exploring the periphery before exploring the center of the environment?
Gosh, that's a long question. I'll just read it. No, we haven't looked at that, but it would be interesting to look at that. I think I'm limited in my expertise as to how to go about looking at that. We have all the X and the Y coordinates and the time, so we definitely have the data that would allow us to do that, but I definitely don't have the programming skills to do that either. So if anyone would like to collaborate, then I would really be interested in looking at different patterns.
Thanks. The next question coming from Nora Newcomb, a very basic question. If you walk around as fast as you can, collecting stars, how long does that take? On another note, why are the Down syndrome and Williams syndrome people so hesitant?
The hesitancy, it might be related to confidence. So one of the studies that we ran in the same project was looking at anxiety, and people with Williams syndrome and Down syndrome have really high navigation anxiety, partly due to lack of experience, partly because they have high levels of clinical anxiety anyway. So that might be to do with their hesitancy. I don't think it's anything to do with their ability to use the computer, because we had a kind of corridor task where we made sure that they could kind of navigate themselves around. And if you whiz around as fast as you can collecting the stars, how long does it take? I'm going to try that this afternoon as soon as I get off. But clearly you can do it in less than the 3 minutes, but quite how quickly you can do it, I'm not sure.
Okay, we have time for one more question. The last one from Michael Hamburger. If I heard you correctly, you said at the beginning that most of these children are not allowed to go out by themselves. I wonder how much this might have impacted their task performance, as they have very little real-world navigation training. Or to flip it around, could one potentially train these children to become better navigators?
Yeah, I completely agree with that. And I think it aligns very nicely with the Beats paper, where they showed that if people have not been allowed out as much, then they are more reliant on a less sophisticated strategy. There's also a paper by Ben Shaw, where he looked at what he called child independent mobility across 18 different countries, and showed a relationship between child independent mobility and even things like PISA scores. So I think to flip it around, you can train people to become better navigators. When I speak to parents about this, a lot of the limitation is not just their spatial and cognitive skills, it's their social vulnerability. So particularly for people with Williams syndrome, they're very overly friendly, and both groups are pretty distracted. So parents would say it's not just the cognitive side of things, it's the fact that they might talk to people or think someone's their friend when they're not. And so we did ask questions in our anxiety paper about travel training and what people have received, and it really varied from 1 hour in a community setting to months of training. And the parents still, the training worked to some extent, but it didn't necessarily cover everything that was needed in terms of understanding money when you get on the tube, or understanding who's a safe person and things like that.
Great, thanks. Thanks again, Emily, for the exciting talk. And then our next speaker is Thomas Wolbers from the 18 and Cognition Research Group at the DZME Magdeburg. So Thomas' work is focused on the basic mechanism of human navigation, as well as age-related impact on the navigation network. Today, he will tell us about mechanisms and consequences of altered grid coding in human aging. I will hand over the stage to you, Thomas.
There you go. Thanks, Rujing. Can you see the slide?
Yes.
Okay, perfect. So as you might notice, I just changed the title into altered entorhinal and hippocampal coding, because I'm not going to talk exclusively about grid coding. So yeah, thanks everyone for tuning in and joining this session. So I'm going to see if I can turn this into a laser pointer as well. There you go. Okay, so what I'm going to do today is try to cover 3 topics. First, I'll work on grid cell coding and path integration in human aging. Then I'm going to talk briefly about recent data on altered exploration of environmental boundaries. And then the final part will look more at hippocampal processing, and in particular on hippocampal hyperexcitability and its consequences for cognitive mapping. So as some of you might be aware of, a number of years ago in 2018, we published a paper where we looked at, okay, how does a grid cell signal, as we can measure it with human fMRI, how does it change in healthy aging? So we had a group of younger and older participants navigate in a virtual environment that you can see here on the left-hand side. And what we observed is mainly that there was a relatively reliable grid cell-like signal in younger adults, and this was absent in older adults. More specifically, it was there, but it was temporally unstable, meaning that the orientation of grids shifted over time. Yeah? And we then also brought the subjects back to the lab and ran a path integration test, because as you've heard from I think Kevin Allen yesterday, that a lot of people in the field think that grid cell, one key computation that grid cells are responsible for is path integration, so computation of position based on incoming self-motion cues. And we did indeed find that the strength of this grid cell-like signal was related to path integration performance in older adults. And this was observed across modalities. So we did it in a purely visual modality, where they would just be seeing optic flow, or in a body-based, where they would be walking blindfolded. And across modalities, we saw a relationship between the strength of this grid cell-like signal and path integration performance. So we wanted to dig a little bit deeper and think about, okay, what is it actually in those older adults that can explain this impaired path integration performance? And of course, the grid cell data that I just showed you might make you think, well, it's all happening within the grid cell circuit itself. And indeed, we do know from a lot of post-mortem studies, but also from PET studies using tau-sensitive tracers, that, for example, in healthy aging, so these are data from healthy older adults, you already see a nice buildup of tau pathology, both in entorhinal cortex and also in parts of the hippocampus. And here there's a correlation with episodic memory strength, even though these people do not have Alzheimer's disease. So there's definitely pathology emerging already in healthy aging. However, as we heard from Yuri Agarwal, older adults often also show deficits in motion perception. Yeah, so obviously vestibular degradation of vestibular processing will impact on your ability to sense translation and rotational self-motion. But likewise, here's data showing that older adults also, or the older people get, they also show reduced sensitivity to visual speed and visual angle. So also visual motion processing seems to be impaired. But of course, we also know there's a lot of data about behavioral neuroimaging, and here's some monkey data showing that aging is often also associated with strong reduction in working memory. So we look at persistent firing here, as we had yesterday from Motoharu, and where, for example, prefrontal cortex, we typically see very nice persistent firing that is thought to keep working memory traces active over some delay periods. And this persistent firing is often drastically reduced in old age. So there are multiple factors, multiple processes that could explain this reduced navigation, path integration behavior, and which could also impact on grid-like coding. So in order to get at that, what we did is we ran a study in which we did a path integration task that looked as follows. So as you can see here, participants would be guided while being blindfolded. So this actually, they were an HMD, but the HMD was black, didn't show anything. So they basically would be walking blindfolded. They were guided by the experimenter. And you see here typical trajectory. So here's the start point, and they would be led along this circuitous trajectory until this endpoint here. And what we did is that along the way at multiple intermediate stopping points, we would stop them, and then they would be asked to turn towards the start location and also estimate the distance to this starting location. So we made those experimental choices a curved path because we wanted to avoid categorical biases, because there's work, for example, people like Dan Montello showing that people have a tendency to regularize turns. So if you do like a standard triangle completion task, and you rotate someone for, say, 70 degrees, they often perceive this as being 90 degrees. And likewise, what we did is we had those intermediate stopping points, so not just one stopping point at the end, but multiple intermediate stopping points. And the main aim here was to basically increase the number of data points that we would be getting from a single trial. We then teamed up with Ida Fied, who's now at MIT, and Ingmar Kanitschneider, and they basically developed a computation model with the aim of trying to decompose performance into multiple sources of error. So it's really try to get an idea, okay, what are the predominant error sources that can explain performance in this task? So you can, and what they included here is like memory decay. So I've talked about working memory, so forgetting of previous location estimates. We had a velocity gain bias on systematic over or underestimation of self-motion speed, additive bias. And then there's internal noise. So there could be like an accumulated noise within the path integrator itself. And then we also included reporting noise, because you might have a very precise estimate of your location, but the process of reading out that estimate and turning it into some kind of motor response could be noisy in itself. So on the left-hand side here, you see performance across those different stopping points, in blue from younger adults, in orange from the older adults. And of course, what you would expect is that error accumulates over stopping points. So the further you've walked, the larger your error gets. But also we see a standard age effect here, and that older adults perform worse, show larger path integration errors, which is kind of what other people have observed as well. We then fitted, as I said, this computational model, and we're able to then compute the relative influence of those different error components to the data. And you see those here. This is across age groups, and you see that memory decay, for example, has a relatively minor influence. There was some intermediate effects of reporting noise. Velocity gain had some influence here, but by far the source of error that contributed the most to explaining this performance was this accumulating internal noise. And very interestingly, this internal noise was also the only of all those error sources here that was different between age groups. So with the older adults showed a stronger internal or larger internal noise compared to younger adults. So it looked, at least from these data, that internal noise was the primary source of error that could also explain performance differences. We then went further and checked, okay, but does this error, this accumulating error, does it accumulate over time or does it accumulate over distance? And the way we could do this is that I told you that there were these intermediate stopping points, but every now and then there were also trials without stopping points, where sort of would just be guided along the entire pathway before then having to point back to start. So in these trials, they covered the exact same distance, but obviously the time it took them to reach the endpoint was much shorter. Yeah? And what we observed here is that, again, across age groups, there was no difference. Yeah? So that whether there was no stopping point or stopping point actually had no influence. And we also validated this with a model fitting approach here. So basically telling us that the path integration error scales in this scenario with distance and not time. Yeah? And this is quite important, because it suggests that obviously that performance is less related to integrator noise, but rather to input coming into the system, which of course could be motion velocity inputs in this case. Oops. Okay. Okay, so let's leave this for the moment. We're going to get back to grid cell coding a little bit later, and we're going to check out boundary coding. So in this scenario here that I've described to you in our study, of course, self-motion cues will play some role, because what subjects had to do is they had to navigate in this environment, learn the locations of 3 virtual objects, and then at retrieval trials go back to those remembered locations. And but of course, there's also salient environmental cues that people can use. So for example, these environmental boundaries, these walls here, that people for sure also will use to remember the location of, in this case, the football. Yeah? We know there's evidence for boundary coding in rodents. Lisa Giacomo mentioned this yesterday, but we have also published a study in 2019 where we found evidence for allocentric direction to boundaries being encoded in the human EC. And Lisa Giacomo has also shown that boundaries might actually help to correct drift in the grid cell system. In the context of aging and neurodegeneration, there's very interesting work from Lukas Kunz, who showed that actually young APOE4 carriers, so being at an increased genetic risk of developing AD, they tend to avoid areas far from boundaries. However, older adults, which has been shown by Nico Schuck, show reduced sensitivity to boundary manipulations. Yeah, so when he had like a circular boundary, it would increase or shrink it. Older adults were less sensitive to those boundary manipulations. So the picture is a bit unclear. And we analyzed those data from this fMRI study and actually looked at the actual exploration patterns. So when people were trying to replace those objects, where did they actually move about? So you see heat map differences here between older and younger adults shown up here. And here we have kind of a summary score. And basically what becomes obvious is that older adults tend to spend more time near those boundaries, which is summarized here, but also that their responses were actually closer to the boundaries. Yeah, so they if they had to replace, for example, this plant stimulus here, they would more often kind of shift the reported location closer to the boundary. Yeah. We then fitted a kind of multilinear regression type of model with multiple predictors just in the older age group, where we try to predict grid cell, this kind of shift in the grid cells orientation that we had observed in that study. And the only parameter in that model that actually was a significant predictor of this shift was actually this boundary score. So meaning, in this case, that the more time people spent navigating in the vicinity of the boundary, and the closer they were to the boundary, the stronger was a shift in grid orientation across sessions. Yeah? So in other words, the grid's boundary navigation was associated with larger grid instability. So this is no evidence for kind of any boundary-based drift correction, at least in the older adults as we tested them. We still don't know exactly what this means. We'll still have ongoing analysis, but it's a very, very striking finding. Okay, so as we all know, there are close interactions between the grid cell circuit and spatial coding in the grid cell circuit and hippocampal place coding, and in particular in the context of cognitive mapping. And when we talk about aging and changes that have been found in the hippocampus and hippocampal cells, obviously a lot of phenomena have been described over the years. So this is a review paper where we had like Carol Barnes joining in and summarizing some of those key findings. And there are multiple phenomena such as loss of synapses, postsynaptic changes, plasticity changes that have been described. And one phenomenon that I want to allude to here, and that I guess will also be covered by Jen Bizon in her upcoming talk, is this phenomenon of hyperexcitability. which has been observed in various hippocampal subfields, so CA1, CA3, and basically meaning that very often when you record from cells in the hippocampus, you observe a much higher firing rate in the aging hippocampus. And this has been linked to, for example, out of field firing, so that place cells would also fire outside their place field. It has been linked to excessive pattern completion. And in humans, it also has been shown, but not in any kind of navigation context. So a bunch of like episodic memory type studies that have reported hyperactivation in the hippocampus. And we were interested to see whether we can actually find evidence for hyperexcitability in the hippocampus in aging as they learn the environment, and B, if it has any relevance for navigational behavior. So the way we did this is that we teamed up with people from the MPI in Tübingen, and they had built up a very nice virtual rendition of the inner city of Tübingen. And we got hold of that model. You see a bird's eye view. So this is just a very small cutout of the inner city. And what would happen is that subjects would be navigating through this inner city from only a first-person perspective. So they'll not be seeing this bird's eye view, but only from a first-person perspective. And they would be actually passively moved. So it was not free exploration, but rather they would see kind of a video. And they would, for example, start here, go to this intersection, go to a dead end, come back, go to the next intersection, and so on and so forth, thereby exploring the entire environment. This was done 8 times, so 8 learning sessions. And after each learning session, we gave them retrieval charts that looked like this. So now you see again the camera here, and but we are occluding any kind of distal landmarks by fog that we simulated here. And then what would happen is that there would be a short movie where the camera would move towards the intersection. It would stop then. And then the subject would be shown with this target image. And this target object could be either this building above here, T1, or this one down there, T2. And what you had to do, if you were a subject, is to now say, okay, and I have to rotate the camera such that this crosshair here faces in the direction of this target building. Even though from this very intersection, and given the fog that we simulate, you cannot see it. So you have to retrieve from memory the kind of allocentric relationship between your current location and the location of this target building. Yeah, so here's overall performance on the left-hand side across, again, healthy older and younger ages. And we see absolute pointing error here on the y-axis, and we have those 8 different learning blocks on the x-axis. And when you look at younger adults, very typically that, of course, in the first session, they haven't really learned very much, but then very rapidly, their pointing error decreases to some kind of floor level, because you always have some basic execution error. So by session 4 overall, they've kind of learned the environment, and then not much more learning happens. When you look at the older age group, you see that they start out again at chance level, but then there's relatively little learning. There's some learning, but not very much, even across 8 sessions. However, the picture is a bit more complex when we look here on this right-hand plot, where we now fitted a learning model to each individual subject's data. And you have in black gray here the young subjects, and in orange the older subjects. And you see that in particular for the older subjects, there's quite a lot of performance variability, meaning that some subjects showed virtually no learning at all. And even by session 8, we're almost hovering around chance level, whereas others were actually able to reach performance level similar to that of younger adults. So this is a very typical finding in aging studies, that the variability increases with age. And we then looked at actually what is actually happening in the hippocampus, and also in particular in retrosplenic cortex / posterior occipital sulcus. So it's kind of medial parietal regions that also have been implicated in those kind of more complex spatial learning tasks. And what we observed in the hippocampus is actually a pattern of responses that we've also seen in previous studies. And not only us, but also other groups have found this, namely that the signal, the hippocampal signal, is kind of indicative of the amount of learning that is occurring in a given session, meaning that in the early phases of the experiment, we see strong hippocampal recruitment when people are still learning a lot, but then over time, when people get better and better, the signal basically drops. Conversely, in medial parietal / retrosplenic cortices, we see the opposite pattern in that the signal actually increases over sessions and kind of mirrors their performance level, in the sense that the better they get, the stronger the signal is that we see here in both structures. So this is we are kind of happy to be able to replicate what we had seen here in previous studies. When we looked at older adults, though, it was different. Yeah? In the sense that in the hippocampus, we see no reliable change of the activity over time, all of those 8 sessions, and also not here in retrosplenic cortex. You know? And this, of course, is interesting because it means that if you find hyper or hypoactivation, at least in the context of learning, depends a lot on the time point at which you look at this, because there's also some conflicting evidence in like the episodic memory literature that some people have observed hyper and others have observed hypoactivation. But here it's like if you look at the early stages of learning, you wouldn't see much hyperactivation, and there might be even a little of hypoactivation. But over time, it's clear that there's a much stronger signal, a constantly stronger signal in the aged hippocampus compared to younger adults. So we wanted to know what might drive this effect here in the hippocampus. And to do this, we then turned to a method called dynamic causal modeling. So basically, we're kind of modeling a dynamic system, which in this case just consists of 2 regions, namely the ones that we looked at here. So those kind of medial parietal region and the hippocampus. And we modeling bidirectional connections between both regions. And then we have the individual level of learning being able to affect the strength of these connections. Yeah? So with the idea that information transfer across both structures could actually be driving, or the efficiency of information transfer could actually be driving the learning effects there. We then feed the navigation periods as kind of input into the system. And in addition, we have inhibitory self-connections within each region, here in PRS and also in hippocampus, to basically, which kind of a standard approach to provide, to eliminate or prohibit runaway excitation occurring here. And what we find is that there was an interaction between age group and the strength of this inhibitory self-connection, meaning that those older adults who showed the poorest learning, that those were the ones where the inhibition was the lowest. Okay, so that was very interesting to us because it was reminiscent of some other human studies, and also the rodent studies suggesting that probably the level of inhibition that we have in the hippocampus could be an important factor to allow the system to build up a stable cognitive map-like representation. So, and with that, I've already reached the end of this presentation. I hope I stayed in time. So to quickly summarize, so path integration deficits in human aging, at least our data suggests that they're predominantly related to multimodal input noise, which could, of course, then drive or lead to this grid instability, as we have seen it. Yeah? So if you have noisy self-motion inputs, that could also, of course, affect the orientation of a grid-like system. We also found that older adults tend to navigate predominantly or more so to near boundaries than young adults do, which is also related to grid instability. And at least in our work, we don't find evidence for boundary-based correction of this grid drift in at least older adults. And finally, the hippocampal hyperexcitability impairs spatial learning, and it could be related to reduced local inhibition. Of course, that's what the DCM data might suggest. It would, of course, be very nice to, for example, complement this with other methods to now look at, for example, GABAergic processing in the hippocampus to see is there actually like a physiological indicator to suggest that this really is local inhibition that is reduced there. Yeah, and with that, I'd like to thank, in particular, the members of my group who all contribute to this work, our collaborators, and various funding sources. Thank you.
Thank you, Thomas, for the interesting talk. So we have one question coming in from Simon Schultz. In mice, there's both hyper and hypoactivity at the cellular level. Could we be seeing both hyper and hypo effects in human fMRI data, the exact balance of which could change with age?
Yeah, so there is, as I briefly mentioned, there is also evidence for hypoactivation in a lot of the aging literature. Yeah, so it's clear that that it's not it's not only hyperactivation that has been observed. Whether we could see that within the same region, within the same task is probably difficult. I guess what is interesting in this context is, for example, these new PET tracers that have been developed, where you can now start to look at synaptic, the integrity of synaptic processing. And there is on the horizon, I guess there are new tracers that could also differentiate between excitatory and inhibitory tracers. And so at some point, I guess it will be possible, but the fMRI evidence definitely has shown both. Sure. So I see a question here from Jan. Should I answer that or? Jing, can you hear me?
Yeah, I just had some problem with my mic system.
Okay.
Yeah. So we have the next question coming from Janvina. When comparing performance for path integration trials with and without stops, could the stops themselves, pointing and reorientation, also affect performance in addition to time?
Absolutely, they definitely could do that. However, keep in mind that in those data that I showed, that the performance between those trials with stopping points and without stopping point didn't really differ. So at least in that setup, we didn't find any obvious difference there. We have newer data though, where we have a more complex task at those intermediate stopping points. So where in addition to pointing to start, they also have to give us a kind of heading response, where the whole response is more complicated and also takes more time. Then actually we start to see that. But in this study, we didn't find it. But of course, it's definitely possible. Oh, well, now questions are coming in.
Yeah, we have more questions coming in. So the next one is from Michael Homberger. Similar to a previous question for the mice, could the participants have counted their steps during the path integration task, which could have helped them estimate the distance?
Yeah, excellent question. Of course, it's a problem. It's a problem that pertains to any path integration study. Like people try to use alternative strategies, such as either counting steps or even like trying to estimate the time. We cannot perfectly exclude it, but I think it's difficult here. It's particularly in this situation, it's very difficult because we have those curved paths. Now, if you have a standard, say, triangle completion task, you have a nicely separated segments. You have translation, and then stops. There's a rotation, stops, another translation. So rotations and translate are nicely separated, and you could think, okay, that people might be trying to count steps to then estimate the distance they covered in each translation. Here, it's much more difficult because as you walk along such a curve, your positional change obviously also depends on the degree of turning angle that you're making. So therefore, we cannot 100% exclude it, but I think it's a relatively unlikely strategy. I remember that we also did some debriefing here and no one really reported it. So I think it's probably an unlikely strategy.
Next question is from Florian. Can you exclude that the bias towards the boundaries in old subjects definitely is a correlative disturbed metrics, but not to overall heuristics?
No, it could. It definitely could be. It could be heuristic. That's for sure. We don't know at this stage. We don't know where it comes from, to be honest. And when you think of Lukas Kunz's work, where they looked at what they called central navigational preference, and this being reduced in those ApoE4 carriers, so they basically thought, okay, when you're in the central navigation arena, you're so far away from boundaries, so that it's very difficult for you to estimate position. So therefore, as a compensatory mechanism, people then try to move towards boundaries. So this could be happening here. But the interesting thing, I think, in this scenario was that if boundaries also serve this function of error correction in the grid cell system, as has been hypothesized by Lisa Giacomo, for example, then we would be expecting that the boundaries should have an opposite effect. So that meaning that the more time and the closer people move towards boundaries, that this should actually reduce the drift in the grid cell system. And we didn't find that. So that's quite interesting. Okay.
We have time for one last question
Okay.
from Christoph. Have you at any chance investigated what influence the reduced inhibition in the hippocampus has on pattern similarity, for example, between different pointing directions?
That's a great question. No, we haven't done that. So I guess this would be related to what I mentioned, the phenomenon of pattern completion. Also, that maybe if you reduce inhibition, you always evoke like a similar pattern, and this could then lead to also maybe similar pointing directions there and similar patterns. No, we have not done this, but it's a great question. Thank you.
Great. Then we will welcome our next speaker. Our next speaker is Jennifer Bizon from University of Florida. Jennifer's research program concerns understanding brain aging and its implications for cognitive function, including memory, executive function, and decision-making. So today she will tell us about targeting age-associated disruptions in excitatory or inhibitory signaling to improving cognition. So the stage is yours, Jennifer.
Sorry. Okay, let me just get this going. Does that look okay to everybody?
Yes.
Perfect. Okay, thank you so much. And I really do want to thank the organizers for letting me speak at this terrific conference with all these fantastic talks. And I'm excited to go after Thomas's talk because obviously the focus of what I want to talk about today is related to changes in excitatory and inhibitory signaling that we have seen be a consistent feature of age-related cognitive decline in our lab. And really a lot of the historical data that has pushed us in certain directions in terms of thinking about how best to approach improving cognitive function in older adults. And so what I'm going to do today is to give you just a brief overview of some of the features of the preclinical rat models that we typically use in our aging studies. As spatial navigation deficits are really among those that are well described in these models. And then I'm going to talk about some of the other cognitive deficits that are prominent in older adults that we can also detect in the aged rats, some of which map onto spatial learning deficits and some of which don't. But I'm going to then spend the last part of my talk showing you some data in which we've linked these different types of cognitive changes to alterations in excitatory and inhibitory signaling dynamics, and conclude with some of the more recent work targeting EI dysregulation as an intervention strategy for improving cognition. So, as I mentioned, we're really interested in understanding the neural mechanisms of cognitive decline. And so we use a rat model to do this. And this slide is really just a basic one to illustrate some common strains that we use in aging research, because this question comes up a lot and provides an overview of the ages we use across the strains. So typically our rats are between 2 and 3 years of age, and our young rats are about 6 months of age. And while there are certainly differences across all of these strains, I have used all of them across my career. And I will argue, hopefully, that there are more similarities than differences. Most of the behavioral deficits that I'll talk about today, we've seen across most of these strains. And a couple tasks, we've only had 2 of the 3 that we've tested, but these are pretty robust effects across the different rat strains. And across all of these rat strains, we don't see frank neural loss in cortical structures, and that's been reported by a number of groups. And in addition, even at very advanced stages, these rats, unless we induce it in some way, don't develop disease-associated pathological features of aging. And that's true of amyloid plaques as well as of neurofibrillary tangles. Despite this lack of pathology in frank neurodegeneration, however, we do see as a very consistent feature of aging cognitive decline, and particularly a decline in spatial abilities. And so the spatial learning deficits can be observed using a standard water maze task, and that's what I'm showing here, in which rats are required to learn and remember the location of a platform across numerous days. And you can see from the small circle on the bottom that typically, I'm sure as this whole group knows, the rat's search path becomes shorter and more reliable or more direct as time goes on, indicative of increased spatial learning ability. If we look at how aged rats do on this task, very typically, young and aged rats perform comparably on trial 1 because neither know where the platform is. But the aged rats, while both young and aged learn, aged rats actually are consistently impaired as a group relative to young. However, as mentioned in the previous talk, we do see considerable individual variability among the aged animals, and such that some of the aged rats perform on a par with young, maintaining performance into advanced stage, where others perform outside this range, demonstrating impairment. And these individual differences are highly reliable, and we've used them very effectively, and many other groups have as well, as a context for examining neurobiological changes, because it's helpful to dissociate those mechanisms that are general features of brain aging from those that may be specifically driving behavioral function. And of course, we know the water maze is dependent on hippocampus, and we also know that these deficits are independent of motoric impairments that also accompany aging. We can dissociate those out. And we also know that these effects don't reflect just a global cognitive decline because they are distinct, for example, from decline in other cognitive domains. And I'll show some of this data a little bit later on tasks in which we've examined prefrontal cortical dependent behavior. So nevertheless, I want to show some additional data from another cognitive assessment that does track pretty closely with spatial navigation impairment within these aged rat study, this aged rat study population. And this is specifically related to perceptual discrimination. And this work draws on findings from non-human primate and humans that have shown the ability to discriminate visual stimuli can decline with age. This has also been referred to in previous talks. But while rats can certainly do visual discriminations, and that's also been talked about here, olfaction is really the primary sensory modality by which rats navigate their environment. And so as a consequence, many rats, or many tasks to assess rodent cognition rely on olfactory cues. And it turns out that early on in my lab, one of the very first studies we were trying to do, we were trying to do something complicated and teach rats something complicated with olfactory cues. And one of the observations was that a subset of our aged rats were impaired in their ability to discriminate between two odors. And that really piqued our interest in this idea that perceptual discrimination abilities may be tightly related to the spatial learning impairment that we've been using for years and years. So, and it turns out, we learned later that the olfactory domain is particularly ideal for asking questions about perceptual discrimination abilities, as olfactory stimuli can be easily varied parametrically. And the example is shown here. These are straight chain alcohols. And the way that you can do this is just by varying the perceptual similarity changes based on the number of carbon atoms on the carbon chain backbone. So for example, propanol versus octanol is separated by 5 carbon atoms, and that will be more perceptually distinct than butanol and heptanol, which is only separated by 3, which will be more distinct than pentanol and hexanol, which only differ by 1 carbon atom. And when we look at this just in young animals, this is performance on a go, no-go odor discrimination task, we see a systematic decrease in accuracy as the stimuli become more similar. And I'll just mention this is the work of Wendy Yoder, who worked in the lab a number of years ago. So how does this type of sensory or perceptual discrimination look in terms of aging? The next slide first shows the discrimination abilities of young and aged animals. In this case, they're discriminating between two structurally unrelated, so very distinct odors. And then the next three show performance within a class that's separated in the way that I described on the previous slide. And you can see that although as they become more similar, both young and aged decrease their accuracy, the aged animals are significantly impaired relative to the young and are disproportionately impaired relative to young. If we look at that in relation to spatial learning, there's actually a very strong relationship. So these are the same data that I just showed you, but now we're averaging this is the percent accuracy across the session. And the aged rats have been separated based on their spatial learning ability. So the aged rats that performed like young in the water maze are shown in light, sort of beige, and the aged spatially impaired are shown in dark orange. And you can see that it's really those same animals that have the spatial impairment that are showing this inability to discriminate the similar olfactory problems. And this is also true. We can see this very well illustrated in an individual correlation as well. So this comes back now to the question that was raised in the previous talk about excitability changes in hippocampus. And so what does this tell us potentially, or this relationship tell us about potential mechanisms in the hippocampus that might be changing in aging? The setup for this was great because one of the things that I think is becoming very clear in the aging literature is it's very difficult to say that there's hyperexcitability versus hypoexcitability, even within a brain region. And so whereas in the previous talk, we heard about CA3 hyperexcitability, which is absolutely true, that is happening in aging. There's also evidence, which I'm actually showing additional work from Carol Barnes' group here, that the dentate gyrus actually becomes hypoactive. And this is work from her group in which rats explored a novel environment, and she looked at immediate early gene expression of ARC expression and showed that ARC induction was actually significantly attenuated in aged animals that were exploring this environment. And so if you think about then the discrimination problem, then in aged animals, not only may you have this increase in pattern completion due to hyperexcitability of CA3, but you also may have a reduction in pattern separation due to hypoactivation of the dentate gyrus. And so when we first started really thinking about how do we target improving these type of deficits, we were really focused on this dentate more than the CA3, thinking that this is what's being reduced. We're not getting that pattern separation that may be necessary for them to discriminate. And so we were actually interested in driving activity in the hippocampus. And to do that, we took the approach of using a GABA-B receptor antagonist. And so the GABA-B receptor is something that we've studied for a while now, and it's a pretty ubiquitously expressed receptor throughout the CNS, and it's a mediator of tonic inhibition. I'm not going to go into all of the complexities of it, but there's actually reasons to think that it's both acts pre and postsynaptically. So it's possible that it could be actually enhancing inhibition in some places and reducing it in others. But when we started these studies, and certainly our goal was simply to ramp up activity in the dentate gyrus. I'll just be honest, that's what we were thinking. So its broad action, though, has made it interest from a cognitive enhancing standpoint, not only in aging, but also in developmental disorders. So we tested the efficacy of the GABA-B receptor antagonist to improve olfactory discrimination in aged animals. And in this study, this experiment was performed. These are all different animals using a within subject counterbalance design, such that all the animals received all doses of the drug. This is just showing first the vehicle, so that you can see that even in this subset of, or in this cohort of animals, we very reliably detect this group of age-impaired animals and age unimpaired animals. And this is the same type of olfactory discrimination task, but here we're just using a trials to criterion cutoff. So this is our vehicle groups. And then if we look at performance following this is a systemic administration of the GABA-B receptor antagonist just prior to the test session, you can see that with the higher doses of the drug, we can completely reverse those discrimination deficits. Now we've gone on to also use this pharmacological approach to test whether we could improve spatial learning. And we've done this in a modified version of the spatial water maze task. And we've explored this version of the task to allow us to try to do repeated testing, and it's challenging to do in the water maze. And so in this task design, the rats receive 3 trials a day, and the platform moves every day. So the first trial is simply telling the rat, where is the platform for today? So it's the information trial. We then do an immediate retention trial. And then we have a variable delay, usually 30 minutes and 2 hours. And then we have a third trial, or the delayed retention trial. And we use the proximity measures on trial 2 and 3. We subtract 2 and 3, and that gives us our index of spatial memory. And the higher the score, the better the memory. So these are animals with no drug, just showing you the effects of age and how this tends to look for us. So the young animals, the aged animals and young animals do both comparably with 30 minutes retention. However, you see the aged animals really start to perform very poorly by the 2 hour, with the 2 hour retention. Now, in our drug study, we used the same GABA-B receptor antagonist. Here, this was delivered directly into the hippocampus. So these animals were cannulated surgically, and then we infused the drug directly into the hippocampus just prior to the test session, also within subject. So the same animals are shown in vehicle and CGP on different test days. The young here are just shown for comparison. And you can see that in this cohort of animals, the aged animals performed quite poorly initially. However, when they're under the condition of the CGP, they really perform much better on this spatial task. So all of this is great, and it does suggest that this could be one approach for improving cognition in aging. However, of course, it's much more complicated for all the reasons that came up in the last talk, which is that this may be useful for maybe enhancing excitation in certain areas that are needed for certain types of cognitive deficits. However, what about in those cases in which there's hyperexcitability? Is that really something that we want to do? And is that going to harm cognitive performance in other types of tasks? And I can give you just an example from our own work that really started to give us pause and thinking about this sort of approach of targeting a single receptor. And this is work now from a PFC-dependent task. We've also explored quite a lot in our lab different aspects of prefrontal cortical-dependent cognitive decline. And what I'm showing you here is that even though the GABA-B receptor is largely maintained in the hippocampus with age, one of the most consistent things we see in the prefrontal cortex is about a 40% reduction of this receptor. So there's a marked decline of these receptors in the PFC. And when we look at that in relation to performance on a PFC-dependent task of cognitive flexibility, we see a very strong association such that loss of the GABA B receptor is actually associated with much worse performance. And so the way to recover the set shifting performance in these animals is actually to take the exact opposite strategy of that I just showed with the GABA B receptor antagonist. And instead, if we stimulate GABA B, the remaining GABA B receptors with the agonist, baclofen, we can improve set shifting performance in aged animals. And this is true if we put it directly into the PFC or if we give it systemically. So this actually poses a significant challenge from the perspective of how we want to treat older adults if they have a plethora of these different these different problems. And really what we want to do is start to think about how can we actually restore some homeostasis both within a brain system or a brain region and then across these brain systems in terms of to really improve, I think, overall cognitive health. And that's really the way that we've started to focus our questions. And these are just a couple of the different approaches that we're now exploring. And this includes things like normalizing HPA axis function, which we know contribute to alterations in inhibitory and excitatory signaling, diet and metabolism. And I'm going to just, by way of example, show you a snippet from some of our electrical stimulation studies, which is one of the areas that we are really pursuing currently. And so this work really started as a collaboration with some of our bioengineering friends at UF. And we got very interested in the vagus nerve. And so for most of you, I'm sure you know that the vagus nerve is both a mixed sensory and motor nerve, and it carries parasympathetic innervation to our organs, but it also has afferents that via the nucleus tractus solitarius actually can regulate neuromodulatory centers and brain activity. And there are a number of clinical applications for vagus nerve stimulation, but one of the ones in which it's been used for the longest is actually in seizure and epilepsy. And this really piqued our interest in this approach. And so we've done quite a bit of work now to be able to implant these cuff electrodes in our old rats. And actually now we can deliver stimulation while animals are performing tasks in our operant chambers. And one of the things, and I'm just going to show this very briefly, but we have shown now that we can actually improve performance with vagus nerve stimulation to a level in a task of cognitive flexibility that we achieve with our GABA B agonist. And that to us was very exciting. Moreover, relevant to this idea of how do we normalize both signatures of overexcitation and overinhibition, we're starting to see with chronic VNS stimulation that several of the signaling proteins that we know or that we reliably see change in age are actually moving towards young levels after only a couple weeks of repeated VNS stimulation. And so one of the things that we now are doing this in a much larger scale way, and so this is very preliminary data, but at least in the PFC, both GABAergic as well as glutamatergic signaling proteins are moving towards young levels of expression, and that's the 0 on this sort of weird graph. And then finally, one last thing to mention about the VNS is that, and this is a collaborative work with Sarah Burke at UF, in that if we give VNS to animals prior to them exploring a novel environment, we can actually increase the activity as measured by ARC expression in the dentate relative to both sham animal as well as a cage control given the same stimulation paradigm. So the next question for us is really whether we can take this initial data with VNS and both improve PFC and hippocampal dependent deficits in our aged animals and ultimately restore some of that EI dysregulation that is very, very consistent across both of those brain regions. And then this slide is just the same one, saying that we're also exploring these other ways of trying to regulate excitatory and inhibitory signaling dysfunction. So with that, I just want to thank my lab group and particularly Barry Setlow and Sarah Burke, who are collaborating on all of this work.
Great. Thanks, Jennifer, for the very exciting talk. I think we can just wait for a couple of seconds for the questions to come in.
Sure.
And also, if the speakers in the panel, if you want to ask a question, you can also raise your hand.
Oops.
Yeah, while we're waiting for the questions to come in, actually, I have a question. I'm curious that you mentioned that the diet can also be used as a way for like intervention or improve the function. So how would you envision that the diet can help in this case?
Sure. So this is collaborative work with Sarah Burke at UF, and she's exploring the ketogenic diet, which also has been used as a treatment for epilepsy. And so her initial work shows that the ketogenic diet can actually also change a number of the signaling proteins that are that we see across animals be changed in both the hippocampus and PFC. So NMDA receptor expression, transporter for glutamate and GABA, and also GABAergic receptors. So that's, and then she's also in our collaboration, we're also testing these in the same types of PFC dependent and hippocampal dependent tasks.
And for this diet to make an effect, how long do you think it will take is it like a lifelong process, or you can actually
unfortunately, right? Nobody wants to stay on the ketogenic diet forever. So actually one of the things that her current award is testing is if you can cycle on and off and what the parameters are for that, because I think that's obviously a very important thing for the long-term feasibility of that approach.
Great, thanks. So we have one question from the audience. Do you think the difference between PFC and hippocampus effect might be reflecting different CAMP levels?
Sure, yeah, that's a great question, and it certainly could be. At least with that, I mean, that's going back to Amy Arnsten's work and many others, I guess, showing that. And certainly that could be the GABA receptor does have effects with cyclic AMP. I think that we have I showed you snippets of data, and we actually have quite a bit of data also from cell EFIS with the collaborator that I have, that in the PFC, we know that there are some, at least in layer 2-3, the pyramidal neurons are under increased tonic inhibition with age. Whereas if he studies, if he does the same types of experiments in CA3, he sees that it's the opposite, right? So they're hyperexcitable, or there's evidence of their hyperexcitability. So I think, maybe it has to do with cyclic AMP on some level, But I think there's also these sort of persistent excitability changes that may be distinct from that, but I think we don't know that. Yeah.
Thanks. So we're also kind of running out of time for the Q&A section, and I would like to thank Jennifer again for your exciting talk. And then we will welcome our last speaker of the session, George Dragoi from the Department of Psychiatry at Yale University. So George's work is focused on deciphering the neuronal codes underlying the formation of internal representations within the hippocampal and neocortical networks. In addition, how neuropsychiatric diseases disrupt such processes. So the title of his talk today is Orientation Selectivity Enhances Generalization and Predictive Coding in the Hippocampus. So the stage is yours, George.
Thank you, Rajing, and thanks the organizer for inviting me. This is a great conference. Looking forward to the poster session and the additional sessions tomorrow. I'm going to share my screen. And I hope you can all see.
Yes, we can see your screen.
Okay,
yes.
So the title of my talk, as Rajing said, is Orientation Selectivity Enhances Generalization and Predictive Coding in the Hippocampus. I'd like to say that my lab is interested in understanding how the brain forms and expresses internally generated representation of the external world. And we do so by studying normal rodents, normal rats primarily, and we do that from a developmental time point into adult. So today I'm going to present data collected from adult long-evans rats. So the motivation for today's talk is, I don't know if you can see everything, is really something that we encounter a lot of times in our lifetime. Say, for instance, you want to look for a place to live and you're visiting, as shown here on the left, a number of properties within a day. And then later in the evening, you're deciding which one to take, and you have to remember the places you visited and how you organize that information. Similarly, you may have your first time visit to a new city and you walk around the city blocks, you went to multiple locations, and then later in the evening, you want to recall what you did and try to organize that information. So then the next day you plan ahead for a new trip. So I'd like to take this opportunity to point out that some of the newer cities have evolved, so newer city plans have evolved in sort of parallel and orthogonal streets, and we will try to understand a little bit later in the talk why that may be advantageous for navigation. So this type of experience raises some type of questions. So I list 3 questions here that were really motivating our work. And this is, by the way, new work published about 2 weeks ago. I encourage you to read additional details there. So the questions are, how do we encode and remember multiple experiences without major interference between these experiences? What is the brain capacity for encoding and remembering multiple experiences? And finally, something that's pretty close to our previous work is, how does the intrinsic function connectivity of the hippocampal network interact with the external stimuli encountered during encoding of multiple experiences? So in order to do that, several years ago, we placed animals on 3 separate linear tracks. It was a U-shaped maze, and then we linearized offline. We linearized the tracks, and they look like that. And we define independent sequence codes that represent each experience on the 3 tracks. So here, I've ordered the place cells based on the location of the peak firing along the trajectory of the animal on each track separately. And you can see here their order based on their sequence of activation. And you can see that the same cells do not form a sequence on the other tracks. However, we can reorder the same cells or the cells from the larger pool of neurons that we collected that day and form a new sequence, which is again unique to those individual tracks. So we think these are independent population codes, neural ensemble codes that can represent, encode, and later consolidate information as it occurred on the three experiences on the three linear tracks. Quite interestingly, we found that, so I'd like to say that the experience that the animal encounters on the tracks is a byproduct of internal function connectivity between neurons that was prior to these experiences, such as shown here by the existence of neuronal sequences of firing that occur during sleep, a brain state when the brain is fairly disconnected from the external world. And these are also naive animals, meaning in this sleep session, they had not visited any single long linear track in their lifetime. So we have sequences of neurons that are organized in time prior to the experience in ways that are later sort of selected and played during the experience. And there is this interplay between this internal organization and this externally driven activity that we're interested in. So at the time, we proposed a model in which functional organization of neurons into neuronal sequences is something similar to a subway map. And then this analogy I like to use a lot, that the subway stations are the neurons identified here by letters, and then the function connectivity or the different colored trains or lines are the trajectories that the neurons are encoding during the experiences. So here, just an example of encoding of linear track 1. Okay. Linear track 2 takes a different trajectory through the network. Linear track 3 takes yet a different one. And then in gray, I'm depicting sort of a generic set of trajectories that exist in the network that have not been played, have not been selected, recruited during the encoding of the three tracks. So we're interested to know what is the capacity of the brain, particularly what is the internal connectivity capacity, such that simply based on that organization of neurons, we will avoid interference between a number of experiences that can occur within the day. So we estimated that capacity based on the fact that during preplay, about 6 to 7% of the events, as we call them, which are multi-unit activity that can take the form of a sequential organization of neural activity that matches one of the trajectories in the future activity. These frames of events occurred in scattered yet clustered forms of activity. They took about 6 to 7% of the whole repertoire of events. So given that activity and given that organization, which appeared to be linear for the three tracks, so 6 to 7% for one track, 14 for two, 21 or so for three, we extrapolated that and provisionally or tentatively suggested that when the animal will be put to explore 15 tracks, that will reach the capacity of the network as currently presented to avoid any interference. So we went on and performed that experiment. We exposed the animals to 15 linear tracks. They were part of two mazes. The two mazes were in two separate rooms, separated either by a door or by an opaque curtain. And they organized as shown here, track 1, track 2, and so on, track 3. And here is a description of the linear track within the arrangement of the two mazes across the two rooms. And you can see that this is essentially a sequence of vertically oriented, so north-south, we call them vertically here, and east-west or horizontally oriented linear tracks. And this is the activity that the animal performed across several sessions. So we were also interested in sleep activity, which will give us information about the internal organization of network in the absence of external stimuli. So we started the experiment by letting the animals sleep for several hours in a sleep box. They're completely naive state, never explored any linear track. Then they run on track 1 for a full session. Then they go back to sleep. And then they run on this top row of of tracks, which are part of maze 1. So tracks 1 through 7, the animals go to sleep again, that's sleep 3, and then they exposed in a different room to tracks 8 to 15, as shown here, and then they go back to sleep again. So this work was performed by data collected by Jeremy Sibyl, and Cafe analyzed the data, and the paper is here if you'd like to consult additional details. So what we uncovered, the network generates 15 maps for the 15 tracks. And here is an example of a single cell, how it fired on the 15 tracks across the 2 mazes. And you can see that there's some activity that is present on some tracks, but not others. We call this phenomenon remapping, and we were interested in how, what are some of the parameters or some of the feature of this remapping? Was it completely random, or it follows some principles, organizational principles of the hippocampal network? So to start, we performed, we used the Bayesian decoder to compute the error of trajectory decoding of the animal based on the activity of ensemble of place cells, either from the same track or from other tracks. And as you can see here, while decoding the activity on a certain track from the place cell activity on the same track, we get a very small error. In this case, was 2 centimeters. However, when we try to decode the trajectory of the animal on track 8, in this case, which is shown here by the dotted line in all the other plots, by using the place cells expressed on different tracks, in this case, track 2, we find that the decoding error increases tremendously. So it's 70 centimeters here. And as you can see, the numbers, they're pretty high. So then we collected all the information on all track pairs, and we compared the decoding error when using the place cells from the current track, which are shown here in blue. They're around 4 or 5 centimeters on average. And then we compare that with the error computed by using activity on other tracks as prior. So that activity is about an order of magnitude, that error is about an order of magnitude higher. So we think that indeed we have 15 maps for the 15 different tracks that the network can generate. On a closer look, we can see a few other things. So for instance, this cell likes to fire on this orientation track. I guess this would be a vertical track, and then fires again on another vertical track and another vertical track, but not so much on orthogonal tracks. So we try to investigate this phenomenon by looking at groups of cells on the linear track. But as you can see here, the activity looks a bit like a mess. It appears to be completely random. However, when we organized the linear tracks based on their orientation, so these will be horizontally oriented tracks, north-south. These will be 2, 3, 5, 7. These will be, yes, 2, 2, 3, 5, 7, and so on for both mazes. We can see that the cell depicted here in blue, and by the way, on top, we show one direction. On the bottom of each line, we show the opposite direction. So this is compressed information. We see here that this cell shown here in blue fired almost exclusively on the horizontally oriented tracks and not on the vertically oriented tracks. And the cell depicted here in red had a similar behavior, but fired only on the vertically oriented track and not so much, almost not at all on the horizontally oriented track. Most of the neurons, however, tended to fire on both tracks and what appeared to be, and then the remapping appeared to be relatively random. So we wanted to quantify this phenomenon, which tentatively we call orientation selectivity in CA1 place cells. And to do that, we built a generalized linear model, and we tested a number of parameters that were guiding the remapping of neurons, population of neurons across different linear tracks. And we found that the only significant and highly contributing factor was the orientation of the linear tracks. So this may give you the impression that by having an orientation specificity, the cells do not remap. So this is not true. Here is a comparison of population place field maps of neurons on the same track exposed twice. So track 1 and track 8 were visited twice. The correlation is about 0.55, close to 0.6. However, when we place the animals on parallel tracks, the correlation, the place map correlation, population place map correlation were much lower. In fact, highly significantly lower, and they were not different from those across what we call analogous tracks, meaning tracks located in the exact same location on the different mazes. So they have same orientation, same arrangement within the maze. So we dissociate the phenomenon of orientation selectivity from the phenomenon of remapping. Moreover, we wanted to quantify this phenomenon because, as I mentioned, not all cells showed this behavior. So here we computed what we call an orientation selectivity index, which is based on the propensity of cells, of neurons, to fire similarly with similar firing rates across parallel versus orthogonal tracks. And we found a population of neurons, about 17% of neurons, that had very high orientation selectivity index. So 1 is the maximum. It's like firing on all tracks of one orientation, but not at all on the orthogonal tracks. So we think these neurons are highly orientation selective. And we use a second parameter, which is the p-value of this orientation selectivity, as compared with 500 shuffles. So we shuffled the orientation of neurons, the orientation of tracks 500 times, and then recompute orientation selectivity. So this second criterion basically separates neurons with very high orientation selectivity index into significant and not significant. So we have about 17% of neurons being highly significant. But I'd like to mention that about 20% of neurons, such as shown here, that do not fire on a lot of tracks to get enough statistical power, but they are orientation selective, meaning they fire only on certain orientation. In this case, one track, but there are others that fire on multiple tracks. And finally, the majority of neurons, about 67%, 63%, would fire on both on tracks of both orientations. Moreover, we're interested to see if this orientation selectivity is specific to mazes. So we looked separately at orientation selective neurons, as shown here, and we find that their preferred orientation on maze 1 was the same as maze 2 in 97% of the cells of those neurons. However, it was 50-50 on the neurons that did not show orientation selectivity. And moreover, the magnitude of orientation selectivity index was similar across the two mazes. So we think of this phenomenon as some sort of generalization across tracks of similar orientation, particularly when it comes to firing rate, neuronal firing rate. So we use principal component analysis to see whether the trajectory of firing rate, population firing rates across different tracks of all orientations cluster. So we color-coded the north-south or vertically oriented tracks in blue, then the multiple, all of the tracks are here from all animals, and then the horizontally oriented in different color. And you can see we could find projections over which the neuronal trajectories across the vertically oriented tracks cluster, and they cluster also away from the horizontally oriented tracks, which are also clustered on themselves. And this is different from the shuffle. So we think we're facing here with a new phenomenon by which hippocampal CA1 putative pyramidal cells, or place cells in the rodent, exhibit orientation selectivity and generalize across tracks of similar orientation. So as I mentioned before, we had animals running, alternating between sleep and run sessions, and this is data from a slightly earlier study. in which we try to understand the contribution of intrinsic function connectivity of neurons as expressed during the naive sleep or sleep in a naive state, how predictive they can be over how the neurons will fire in sequential order run on a novel track that they have not seen or been on before. So in this study, we looked at individual events, meaning multi-unit activity across this pre-run sleep session, and this collection of 76 principal neurons that were recorded at the same time. We just want to focus on a few of them, in particular 2 of them here shown in red, and we show that the cell on the top fires before the cell on the bottom in frames 1, 2, and then again on frame 4. They fire in the opposite order in frame 3, and then in frame 5, only one of the neurons was active. So we looked at all frames, and we computed a probability of one neuron firing after the other. And in this case, just a simple-minded kind of calculation will give you that in 3 out of 5 frames, this is the order in which the neurons fire. So it's a 0.6 probability of neuron on top firing before the one on the bottom. We also were interested in finding the state order dependency. In other words, on how many neurons firing before is the firing of one particular neuron dependent on. So we find that the state order dependency is 1.55, which means that the activity of one neuron seems to depend on 1 to 2 neurons that fire before it. So given that, we build a transition matrix of probabilities of dependency of neuronal order. So basically, you can think of future cell ID being the cell on the bottom and the present cell ID being the cell on the top. There will be an entry somewhere here, but we did that for all cell pairs, and we computed the probabilities of one cell firing after the other. And we used this to build a Markov chain model to predict the neuron order of firing on a linear track here during run, based simply on the neural order that seems to be at higher probability to occur during the sleep. So here I'm trying to show to you that there are 2 factors that guide or participate in neuronal selection as firing in a certain sequence during run on a novel track. The first factor is the one I mentioned. This is the intrinsic function connectivity, such as the one that occurs during sleep. The other one is the response of neurons due to external stimuli that occur during run. So we had some control over the intrinsic function connectivity, meaning we recorded that activity, and we did look, as I shown in the previous slide, at that order, but we had no idea as to what stimuli the network was responding to during run. But we'll come to that towards the end of my talk and see that we've made some contributions here too. So simply based on intrinsic function connectivity during sleep, we were able to show that the probability of predicting this particular neuron sequence that occurred during run was pretty high. And we did that by multiplying the pairwise probabilities between cell pairs that were forming the sequence. So first to second cells had a probability of 0.5, second to third 0.3, and we multiply those, the probability will be 1 to 3 would be 0.15, and so on. So we ended up with a relatively low number, but significant numbers of probabilities. Then we shuffle the order of neurons as they occur during run 1,000,000 times, and we generated what we call this shuffle sequential probability. And then we placed the real run sequence probability as computed from intrinsic function connectivity for the sequence that actually happened during the run. And then we determined its percentile over this 1,000,000, and this is log normal distribution. So with that, since we had 6 animals and 2 directions of running, we were able to compute the probabilities, the percentiles of probabilities of a particular sequence as predicted simply from intrinsic function connectivity as determined during sleep. And to our pleasant surprise was that in all 6 animals, those percentiles were at or above 95th percentile. And in some animals, both directions could be predicted with that high precision. So again, across all animals and all the runs, we could build and then we had multiple runs that the animals took on this track and other tracks. We could estimate that we could determine that these percentiles are heavily shifted to the right, meaning they were exceeding sort of a chance distribution. So we think of this as hippocampal generating predictive codes that will be later be used during the run, and of course intermingled with information from external stimuli to generate these unique sequences during the run. So we came back to the current experiment with the 15 tracks and used the same methodology to determine the contribution of intrinsic organization of neurons as determined during sleep to sequential organization across multiple tracks during the run. And here is again a summary or description of alternative sessions, sleep and run on different tracks, and these are the mazes. And what is interesting that we're able to replicate the existence of predictive codes for all 15 tracks at a group level. And this stems from, if you like, from pre-play, but because this sleep is preceding all the activity. In sleep 2, we found, and this information is summarized here, we found that the probability percentiles were stronger in sleep 2. And we think this is due partly to the fact that the animals have explored one track. So we now have a combination of replay of track 1 and replay for tracks, sorry, it's a mistake here, 2 to 14, although track 1 was used again. And then in sleep 3, we find that replay of tracks 1 through 7 essentially increase the probability percentiles for all the tracks, and this was maintained in sleep 4. So we think there's a plasticity that is induced by the experience that updates these predictive codes, this sort of internal model with information collected during exploration of the linear tracks, and updates this model, which now becomes even stronger predictor of activity. And finally,
George, you have about 1 minute left for
exactly 1 slide left. Thank you, of data. So we try to combine this intrinsic function connectivity information with information that we gather from runs on other tracks, given our discovery of orientation specificity of neurons. And we added this information into the Markov chain model, and we explore the predictability of activity of sequential activity on linear tracks as detected from this combination of sleep and orientation selectivity. And just to guide you, we find that orientation selectivity enhances predictive coding when added to the sleep in sleep 1 and 2. And then we found that enhanced predictive coding is analogous to replay. So if you add information from orientation given by orientation specificity on different tracks to the information from the sleep preceding the experience, then you gain predictive power as if the animal has already explored the linear tracks. And finally, we find that enhanced predictive coding is consistent with generalization, meaning after exploration of the first maze, we don't gain any additional power, predictive power. So in conclusion, I'd like to say that hippocampal place cells exhibit selectivity to spatial context orientation. Orientation selectivity enhances generalization of representation for multiple environments, and then also generative predictive codes. And finally, to give you a hint as how we can envision this while we explore, city blocks. Here is an example, a real example of a orientation selective place cell in the rodent, and you can see it fires on tracks of parallel orientation, but not on orthogonal one. And I'd like to thank my lab for great daily work, particularly Cafe and Jeremy, who contributed significantly to this work, my host departments and the funding agencies. And of course to you for sticking around for this late hour.
Great, thank you, George, for the talk. So we have a question coming from the audience. The first question from Kate Jeffrey about your very interesting orientation selective cells. Were they more likely also to be direction sensitive, i.e. unidirectional?
So we compare that. It's in the full article, and we find that orientation selectivity exceeds the directional selectivity. There are a number of other factors that seem to have been exceeded by this orientation selectivity phenomenon. That's why we prefer to present it as the primary phenomenon that guides this similarity or of activity across parallel tracks.
One question from Kevin Allen. Do you think the pre-play events would also include CA1 and entorhinal ensembles, or do you think the key connectivity is within the hippocampus?
The particular analysis that we did relies on cell pairs in this case. So lesions of the entorhinal cortex have shown to largely preserve short motifs of sequential motifs in the CA1. So it is hard to speculate beyond that. We think it's a combination of intrinsic CA1 function connectivity that could be guided by CA3, but also we think entorhinal cortex does play a role, particularly in updating the model as shown. So during the run, I think the entorhinal activity does contribute in a major way.
Great, thank you. So since we're towards the end of this session and there are more questions coming in, I suggest that we can continue the discussions on Gathered Town. So thank you, George, again for the interesting talk. So yeah, thank you to all the speakers for the fascinating talks and also to all the attendees for your participation.